So one of the most fascinating things about the human race is our brain. It's the organ that makes us think, fall in love, laugh at jokes. But its uniqueness also makes it very difficult to study in an experimental setting. Today, we see how we can use clinical tissue as a way to study the human brain, how it develops, and region-specific molecular properties that perhaps make us human. So sit back and enjoy while we take you a tour of how we use the mass spectrometry-based proteomics to study the global proteome of different regions of the developing human brain. So hi everyone, welcome back. Today we're going to be talking about mass spectrometry and how my lab is using it to study neurobiology and various types of neuropathologies. So this is the brain. This is arguably the most interesting organ of our human body. It's what allows us to stay creative, fall in love, contemplate the meaning of life, and really sets us apart from the other life forms on planet Earth. Now, while it may be interesting to study for these reasons, it also makes it exquisitely difficult to do research on, because, of course, the unique features make studying the human brain very difficult in lower mammals such as monkeys and mice that have a much more simplified anatomy, as this picture shows, and thus may be able to carry out some of the kind of common biological functions that we share, but perhaps some of the more interesting ones will be absent in these model organisms. One of the other challenging components of the human brain that makes it difficult to study is that, in fact, it's very heterogeneous from region to region. This is a Nissl stain taken from the Allen brain map, and you can see here that parts of this section of the brain that are cut coronally are purple, which are the areas of the gray matter that house the neurons, whereas the lighter stained sections, the white areas, are the myelin and white matter. And as you can see, this makes it exquisitely difficult to study, because you can't just study the brain as one entity, but you have to study it based on its regional complexity and identity. And thus, it becomes a much more exponential task to try to understand the unique biologies of these different lobes that, of course, have inherently different functions. And so how this has been addressed by some people is here are two papers that decided that in order to thus study the brain, we must study its spatial, so how it differs from region to region, and also its temporal changes, how regions change with age and development. And thus, what these studies have done here is they use transcriptional profiles to try to understand what the molecular differences is between different regions of the brain and during different stages of our lives. And I think this is a really good example of how you can start using human tissue to try to unravel some of the unique features of these different parts of the brain, and perhaps try to see if they have an indication of these special functions that humans have. So we've been motivated by these papers. These are papers that really have shaped the way we think about studying the brain in our lab. And what we're particularly focused on is understanding the phenotypic aspects of the human brain. And what that means is that we're interested in studying protein and then further downstream modifications that occur in biology. This is kind of a one way to conceptualize the different molecular components of our tissue, And of course, most of the information is stored in the DNA. This, of course, needs to be transcribed to RNA in order to provide instructions for the different regions to carry out different functions through a process called transcription. And then RNA is then translated into proteins, which is subsequently modified by glycopeptides and phosphorylation events to really drive activity of these protein structures. These protein structures can, of course, form protein-protein interactions, which are required to form pathways and networks that really drive the phenotype and the traits that we were able to conceptualize when we look at the brain, and also things like our personality. And we're interested really in understanding how the proteins vary in these different regions and through time. And we call this we call this area phenomics, because it's really looking at the phenotypic characteristics of tissue. So as a pathologist, the way I do this is by looking at the H&E morphology of tissue, and also use what's called immunohistochemical staining to look at individual proteins and their expression and distribution at the single cell level. And you can see that these are very, very powerful approaches to understand how structure and function correlate with specific phenotypes. Now, one of the limitations, of course, of these is that morphology is very qualitative, and immunohistochemical staining really only allows you to look at one or two proteins at a time, which, of course, is very powerful, but somewhat limited in its scalability. So the areas of the lab, as you've probably seen from other lectures, is an interest in kind of transitioning this into more global approaches. So for morphometry, we try to use AI and deep learning to try to automate pattern recognition tasks. And what this allows us to do is look at specific patterns across hundreds or even thousands of slides in a relatively automated way. And I think that is really the future of looking to see how subtle changes in histological patterns may correlate with specific phenotypes that we see of interest. Immunohistochemically, we're trying to use a technology known as mass spectrometry to look at the global protein patterns rather than having to look at individual proteins one at a time. And thus, this is another kind of technology that really looks to scale this powerful technique and allow us to monitor protein patterns in tissue compartments. Now, why do we want to do this? Why not look at the RNA levels, which the technologies are quite mature, and try to infer protein levels based on the mRNA abundances? And for the simple reason that I think this profound quote is quite fitting, is that predictions are very difficult, especially about the future. And of course, this is a quote that I think we can all relate to. We can relate to this when we watch the Weather Network. Even predicting what tomorrow's weather will be is in fact very, very difficult. And this has been the experience also in biology. Even though RNA is the precursor of which proteins get translated, the actual abundance of these of the protein given a specific mRNA molecule is in fact quite poor in multiple studies where they try to correlate the relative abundance of RNA and protein. You can see here these Pearson correlation coefficients. The two highlighted here are 0.2 and 0.18. Of course, the perfect correlation would be 1. No correlation would be 0. And so this is very close to a correlation where we can't really predict levels of protein based on RNA levels. And of course, this changes based on the specific situation and question you're going to ask. But suffice to say, we do not have a sufficient we do not have sufficient information at the RNA level to really understand the networks that are in fact occurring. And there's been a number of papers over the last few years that have really highlighted this detail. This is one of the many papers by the CPTAC group that tries to integrate proteomics and phosphoproteomics into existing genomic and transcriptomic data from the TCGA network. And what they found is that across multiple different tumor types, the protein's levels only modestly correlate with DNA and RNA measurements. That R value, again, is the Pearson correlation, showing that the relative correlation of mRNA and protein abundance is in fact quite poor. And what this means is that you can have copy number alterations, so significant changes in the genomic code that can lead to mRNA differences at the cis level. So RNA differences at the areas where the copy number alterations have occurred. But when you look at the protein levels, most of them occur at trans loci to the copy number alterations. So they occur in distant areas of the genome that could not have been predicted based on these copy number alterations. So why is this important? Is because when you look at on protein networks and pathways, when you try to predict what pathway a cell is utilizing to carry out a specific biological function, you get in fact very different biological functions based on whether you use RNA and protein. So here, for example, using these pathway-based groupings, the protein-based approach is going to highlight different patterns such as invasion, cell migration, immune regulation, which may not have been picked up using mRNA values. And of course, that's very concerning given the importance of these pathways for resistance and kind of emerging therapies. And really, the tiebreaker, in my opinion, is that when you look at prognostic signatures, as you see here, the ability to predict which patients will have a more indolent course is far superior when using proteomics compared to transcriptomics, at least in this example of ovarian cancer. These are just some more images from this seminal paper, just highlighting that. Again, highlighting the fact that while it's true that there is a positive correlation between RNA and protein, the actual median correlation between these different pathways is, again, in fact, quite modest. And when you do the pathway analysis here, you can see that you're going to nominate different pathways to target in precision medicine efforts based on whether you're using copy number alterations, transcripts, protein, and phosphoprotein. So you can see here there's a number of pathways that are enriched at the phosphoprotein level that you would not have otherwise predicted if you were to focus on the pathways that were highest at the transcriptional level. So significant implications for precision medicine based efforts. So hopefully that explains the motivation behind trying to update some of the transcriptional maps of the human brain with some proteomic information. And that's really one of the goals of our labs. This is one of our kind of manuscripts highlighting how our approach works. We used a very similar approach to those seminal transcriptional papers where we could create these slides. We can stain them with specific stains like hematoxylin and eosin to allow us to visualize the set architecture. We could delineate those and micro dissect those to purify for specific regions to kind of overcome this regional heterogeneity. We can extract proteins from these regions, digest them using trypsin into these shorter peptides, which can then be separated by a liquid chromatography and run through a tandem mass spectrometer, which will use these kind of spectral fingerprints to identify what proteins are found in the different compartments that have been microdissected and analyzed. Now, in the lab, we call this workflow prompt, or proteomic molecular profiling of tissue. And one thing we wanted to do is really outfit it on formalin-fixed tissue, and you'll see why that's an advantage. Usually, tissue in the pathology department is either stored as frozen tissue, which is traditionally been better for molecular analysis, or as formalin-fixed tissue, which is really nice for looking at anatomy. But one of the other kind of key things about formalin-fixed tissue is that, in fact, it can be stored for long periods of time. And of course, because you have a corresponding slide, you get a very rich understanding of what each block contains in it. Now, because we can store these for quite some time, as you can see here, we can use that to our advantage to select very, very specific cases that fit our scientific questions. And so if we could optimize our mass spectrometry-based proteomics for this types of tissue, we could ask very, very unique questions, as we'll see, that would otherwise not be possible with frozen material. So our workflow, again, is select cases of interest, microdissect out the areas such as the cortex that we want to study, and then do global proteomics to understand the protein patterns, and then verify these with more traditional proteomic techniques such as immunohistochemistry. So this particular talk is focusing on using this technology to study neurodevelopment. So I want to take a few minutes to provide a review of this process. So hopefully it allows you to put our data in better context. So as we know, the brain forms mostly during the first 40 weeks of in utero life, while the baby is still within the mother's womb, and of course continues to develop afterwards. And really the brain really begins to form as this neuroectodermal structure when there's a three-layered embryonic structure. You have your neuroectoderm, your mesoderm, and your endoderm, and it's the ectoderm that's going to form your skin. But part of this ectoderm is what's called the neuroectoderm, which is responsible for forming your central nervous system. And what happens around week 4 post-conception is the middle component, this neuroectoderm, will fold backwards upon itself to create this neural tube around week 4. And that is really this single layer structure is what's going to form the central nervous system. And this process is called neurulation. So once formed around week 4, this will undergo a massive proliferation potential where this single layer structure will divide these stem cells, these Nestin positive, SOX2 positive stem cells will divide quickly to form this multi-layered structure known as the subventricular zone, which of course will then undergo a process called neurogenesis. During the first half of gestation, around week 12, these neural precursors will form neurons and migrate out of this ventricular zone into this kind of peripheral area known as the cortical plate, which is going to be your future cortex. The subplate is a structure that helps coordinate axon connections from region to region. The intermediate zone is going to be the future white matter, so there's a lot of axonal processes in there. And here is your ventricular zone or subventricular zone that carries these stem cells that arose from this neural tube. And histologically, you can see here, you get different cellularities that allow you to see these under the microscope. Again, your ventricular zone, your intermediate zone, your subplate, and your cortex, which create this very nice linear arrangement of immature to maturing to mature cells. So once this neural migration has ceased, which usually occurs in the first half of in utero life, you basically have all the neurons in their correct location for the rest of your life, which is an amazing thing to think about. During the second half of gestation, the molecular program in this ventricular zone will switch and undergo a gliogenic program. So these neural stem cells will change from trying to create more neurons to focusing on developing astrocytes and oligodendrocytes that will populate the remainder of your brain and provide support for your neuronal cells. And further in development, your neurons will again begin to mature, form synaptic connections with one another, and of course extend their dendrites to help form these circuits that provide consciousness and the ability for us to learn and carry out the functions that we know the brain is capable of. Myelination occurs again in the later aspects of neurodevelopment, also synaptic pruning. So this idea that some connections that are not useful for us will degenerate and allow the stronger connections to really dominate how the brain is wired. And all this stuff, of course, leads to the final familiar structure that we're familiar with, which is the kind of mature human brain. Now, as you can imagine, this is a very complicated process, so a lot of things can go wrong at any one of these steps, and a number of diseases have been implicated and pinpointed to one of these amazing kind of neurodevelopmental milestones. So microcephaly, small brains are thought to be a defect in neurogenesis. There's some autism-related disorders that are thought to be due to abnormal synapse formation, and of course, even some adult-onset disorders like schizophrenia are really thought to be. There's a growing kind of consideration that these, in fact, may be neurodevelopmental disorders that have origins early on in life. So the idea is that if we can understand how the brain develops and the molecular machinery that drives these processes, perhaps we could better understand and begin to reduce the burden of some of these very challenging disorders in human life. Now, how do we study this dynamic process using human static tissue? Well, as you'll see, our approach has been to use the advantage of this formalin-fixed paraffin-embedded approach to get fetal brains that have died of other causes from our autopsy archives going many, many years back, and find normal brains from fetuses that had heart abnormalities, where they didn't survive the in utero developmental process, but otherwise had normal brains. We can now study these during these different developmental milestones and see how those protein patterns change through their developmental life. And hopefully this will provide clues about the molecular machinery driving these important processes. So that's exactly what we did. So here you can see the half of a brain of a human fetal brain. So here you have your ventricular zone. Here you have this this precursor compartment called the subventricular zone. Your intermediate zone, again, you have these migrating neurons in there, but is really going to form the future white matter. The subplate, again, is a important structure that coordinates neural connections. It's very large in utero life, but shrinks to form layer 6 as we age. And of course, the cortex is going to be the future gray matter, where the neurons migrate and mature. So we took brains from 16 weeks to 36 weeks, so pretty much half of development, but really capturing all those specific neurodevelopment milestones we've discussed. and we dissected out these regions of interest that have stem cell, mature neurons, and other kind of transitory neural elements, digested them using trypsin to form these tryptic peptides, which allow us to study them using this instrument called the mass spectrometer. Now, although we've created a very simple model here using this fetal brain, of course, these tissues are in fact quite heterogeneous. There's vascular tissue that provides blood and oxygen to these different regions, and thus, of course, these protein profiles that we generate are going to have some confounding variables. So to combat this, we also teamed up with James Ellis's lab from Sick Kids to create a kind of in vitro surrogate of this process where we could generate human neural precursors from induced pluripotent stem cells, so taking someone's skin and converting it by reprogramming into a neural precursor cell, and then directing it down a neuronal differentiation that uses a depletion strategy to remove non-neural cells and have a kind of a purified neural population that would correlate with the kind of cortical tissue of the fetal brain. And of course, this neural precursor culture would provide a purified correlate with our ventricular zone. So we did these two experiments in parallel, and what you can see using what's called shotgun proteomics, we could identify about 2,500 proteins per region, regardless of whether they were the in vitro cultures or the human tissue elements. And now what we can start to do is start to make sense of these different processes. So as a quality control, we can look at we can use these things called Venn diagrams to see what proteins were found in these different regions, and you can see that many of the proteins, so out of the 3,000 proteins we identified, many of them were in fact co-localized to a specific region, were in common with across the different regions, and then there was some proteins that were more specific to a specific region. So these would be interesting to study to understand the unique biology that protein plays in regional biology of that region. We saw a similar kind of proportion in the in vitro cultures between induced pluripotent stem cells, neural precursors, and neurons. And again, when we kind of look at both data sets together, we identified about 3,800 proteins of the human central nervous system at this early stage. So very, very useful data in our opinion to understand human cell processes. So another thing we can do is we can look at the biological functions of these proteins to see if we're biasing our results using this formalin-fixed tissue to a specific biological function. And in fact, you can see that we cover a lot of different biological functions, and the relative proportion of them is equal between our formalin-fixed tissue and our in vitro cultures. So we thought that this, in fact, validates that our formalin-fixed approach is in fact quite robust. Similarly here, we see that the cellular compartments, so the proteins are distributed throughout different aspects of the cellular compartment, again, really highlighting the fact that we're capturing a wide range of biology in this approach. So we wanted to see first if we could kind of validate some of these results before we can start making some questions about if we can discover unique biology. We wanted to first show that our system was in fact robust. So we focused first on the in vitro assay that was, of course, a much more controlled environment to look at protein patterns. And we wanted to see if the protein patterns identified in the induced pluripotent stem cells were different than the neural precursors and neurons that we subsequently differentiated these cells into. And what you can see by these Pearson correlations, you can see that although the correlations between these different specimens, these replicates, are quite high, as you go outside of these, if you compare them outside of their unique identity, the Pearson correlation in fact decreases. So that's a good indication that through maturation, the correlation of the proteins does in fact quite change. And you can visually also see this using what's called the principal component analysis, that shows you the kind of the distance between each point tells you the relative similarity of the overall proteome. And you can see that these different cell states have very, very unique proteomes that transition from induced pluripotent to neural precursor to more differentiated neurons in a very gradual manner. So we can now study these what these changes are and make inferences about the biology of these cells. Now, what you can start to do once you have these proteomes is you can identify specific proteins, individual proteins that are different between the different compartments. So that's what this volcano plot is trying to depict here. It's looking at neurons and neural precursor cells and identifying proteins that may be enriched in neurons versus precursors. And you, of course, can see interesting proteins like DCX, MAP2. These are neuronal proteins, kind of verifying that this is, in fact, working quite nicely. But of course, and then here's nestin. This is a precursor marker, vimentin, again showing that these markers are in fact enriched in the group that we believe they're enriched. And once you develop enough confidence in this types of data, you can now look for unique proteins that are enriched in one compartment or the other. And what we're doing in this table here is we're looking for proteins enriched in a specific cell type, but also importantly, proteins that are expressed on the cell membrane, allowing potentially to use technologies like fluorescent activated cell sorting to sort out these populations, which is of interest for a number of labs looking to understand stem cell biology. So now that we're confident that this works in the very controlled in vitro environment, can we apply this to more clinically oriented tissue specimens? So again, we took our fetal brains, 8 spanning this gestational week 16 to 36 time frame, and we dissected out these different regions and quantified the proteome of these 30 plus samples. And what you can see here, based on this kind of heat map, which shows you the intensity of a specific protein relative to the specimen it came from, you see that by grouping the samples based on the intensity of the most differentially expressed proteins, you get very nice clustering of ventricular zone areas with one another. You get cortical areas grouping together, suggesting that these different regions are defined by very specific proteins that are turned on in one region but are off in the other. Now, what you can see too is that based on this kind of family tree, you can see how the individual compartments change with time. So you can see here ventricular zones from very early fetuses grouped together, while ventricular zones from later fetuses, 36 and 26, actually group more distantly. So not only are there spatial differences, but there also seems to be temporal differences based on this heat map. So let's again try to build some confidence in our data so we can start to ask novel questions with confidence. So one way we can do this is we can group proteins based on the cellular compartment they're from, the biological process that they're involved in, and also their molecular function, and graph that on this 45-degree angle to see if there's specific processes enriched in the ventricular zone, which would be on this side of the diagonal line, or processes enriched in the cortex, which would be below this diagonal line. And you can see that within the ventricular zone, this precursor compartment, we get a lot of DNA replication and cell replication proteins. Whereas below this line here, we get more synaptic related processes such as ion channels and synaptic vesicle formation. Again, showing that in fact we're picking up many of the pathways and processes that we're expecting to find in these different regions. So again, that would be at the global level. We can actually look at specific candidates that we know should be enriched in certain areas. So proteins like vimentin and nestin should be enriched in the ventricular zone, and you can see here these negative values depict that in fact they're increased multiple times in the ventricular zone compared to the other compartments of the brain. In the intermediate zone, this area that's going to be the future white matter, we see an enrichment of these neurofilament proteins, both the heavy, light, and heavy, medium, and light chain neurofilament proteins. Again, really proving that this is an area where there's already axons being formed. In the cortex, again, you can see here, these are specific transcription factors found in different layers of the cortex, which kind of provide cell identity and specification for different neuronal types. And again, they're enriched in the cortex compared to the other regions that don't really house neurons, and thus you wouldn't expect them to have neuron-specific transcription factors. This here is just showing just how well the proteomic data correlates with the immunohistochemical data. Of course, immunohistochemistry is a technique that we're all very familiar with in pathology. So we want to build confidence that what we're seeing by these label-free quantification values from the mass spectrometer, in fact, correlate nicely with the kind of gold standard or immunohistochemical staining. So these are ventricular zones from different gestational weeks, and what we see is that in fact this is the H&E, this is the kind of hematoxin eosin stain section, where we can see that these look fairly similar, but when you stain these with GFAP, which is a glial fibrillary acidic protein, you see that this is really turned off until later aspects of neurodevelopment, which, as we said, is when you have this gliogenic program that is turned on in the second half of gestation. Conversely, nestin is very strong early in development, and as you can see, reduces with time. And this is exactly what we saw using these mass spectrometry-based methods. So you can see nestin goes down with gestational age, whereas GFAP comes up. And of course, this is almost a one-to-one correspondence between these two different proteomic techniques, really highlighting the power of mass spectrometry-based proteomics to do this at a global level. This is calretin. This is another protein found in neurons. And as you can see early on, when the neurons have migrated, these blue dots here, but have yet to mature, they're relatively negative for this specific protein. And as the cortex matures later in life, you can see that this protein is significantly in higher values later in life. And you can see that that's exactly what we see with these LFQ values. We get almost a one-to-one correspondence where it's relatively low. It starts to pick up here. And again, we get very, very high values of this protein through development. So now we've hopefully developed confidence in the system, so we can look for perhaps unique proteins that are uncharacterized, haven't been reported in brain development, and see if our approach can in fact identify some interesting candidates for further study. And the ones we chose to pick based on this criteria were the cellular retinoic acid binding protein 1, or CRAB-P1, and filament C. These are proteins really that have been poorly characterized in the brain, so we thought it'd be a good opportunity to see if we can validate some of these novel proteins that we've identified. So we can do this with immunohistochemistry. The beauty of immunohistochemistry is that we can analyze these proteins at the single cell level. So we stain these fetal brains here with filament C and CRAB-P1, and you can see that they have very, very distinct staining patterns. filament C really lined this ventricular lining here, and interestingly only lined the ventricular lining of the medial and lateral ganglionic eminence, and really then was absent from the this subventricular zone as you move to the corpus callosum here, and this pattern matched nicely with SOX2. Again, SOX2 is a stem cell marker, and we couldn't find filament C in the cortex. So we thought that this could perhaps be a very, very specific stem cell marker that could provide new insight into stem cell biological processes and cancer. So that was one interesting marker we found. The other one, the cellular retinoic acid binding protein, again had a very similar, or interestingly similar staining pattern, where instead it was found scattered throughout the ventricular zone in rare cells, and then in the medial ganglionic eminence we found these cords of hyperstaining cells, which seem to form this kind of migratory stream. We also found strong staining in the striatum and then the Cajal-Retzius cells. So again, a protein enriched in the ventricular zone with a very, very specific function that warrants a future investigation. Now, one of the challenges with doing this in tissue is that in fact it's open to interpretation. So many of us thought in the lab that filament C was in fact staining the processes of the neural stem cells that reach out into the ventricle, whereas the alternative hypothesis, of course, brought up by others, was that it was in fact staining the ependymal cells that line this juncture. So this is in fact two reasonable hypotheses. So how are we going to resolve that? Well, we turned our attention to the in vitro system. So we gathered that if filament C is a neural stem cell protein or a neural precursor protein, we would see it activated in the neural precursor compartment, and then this would be turned off while the cells differentiate. And in fact, you can see that was the case. So what we have here, again, is evidence that this filament C is a protein that is co-expressed by Nestin-positive cells or precursors and is down-regulated during development. So hopefully I showed you how we can use proteomics to study this very dynamic and complex process and identify novel proteins that may be playing a role in driving some of these interesting processes. Now, of course, you can do the exact same thing with different neurodevelopmental diseases now that are implicated to occur in specific time points and compartments of the brain. And this is really the advantage of the formalin-fixed tissue, is that you can retrospectively go and find cases that have been diagnosed with a specific disorder and try to understand how the protein changes could provide insight into the biology of the disease. So as you can see here, microcephaly is thought to be a derangement of neurogenesis. And so this was actually one of the diseases that was in the media at the time that we were preparing this manuscript. So we thought it would be an interesting disease to study using our technology. And it was in the news because of the Zika virus, I guess, pandemic, where this virus that was found in largely Caribbean countries caused microcephaly. So the question was, how was this virus causing microcephaly? And many hypotheses were brought forth about whether it was actually causing neural precursor cell death, or if it was in fact causing premature maturation. And so to address these alternate hypotheses, we didn't have brains from Zika virus available to us at the time, but we had access to a much more common cause of fetal viral encephalitis or microcephaly, which is the cytomegalovirus, which again causes a similar phenotype, but it's much more common. And really, the hypothesis at the time was, in fact, that it infects these proliferating stem cells and leads to microcephaly by causing death of these stem cells required to populate the brain's neural compartment. So to test this, we went into our archives. We took 3 brains from normal mid-gestation fetal brains, and we compared them to CMV infected brains. And again, using this kind of regional microdissection approach, we dissected out tissue from the ventricular zone, intermediate zone, and cortex of these different brains, and wanted to compare the proteomes of normal, relatively normal fetal brains to these CMV infected brains. What we found is, again, across all of the regions dissected, when we look at the kind of pathway enrichment, we found many processes involved in immune regulation, interferon activation, immune response to foreign agents, as you would expect, such as here antigen presentation, which was expected, but importantly, it was most enriched in the ventricular zone, which of course is interesting because that is where the CMV virus is thought to be activated. Now, more interestingly, what we found also is that there was a substantial upregulation of proteins involved in axonogenesis, neuron projection, dendritic spine morphogenesis. These are processes that we should be seeing in the cortical plate, and as we saw before, are now being ectopically found in the ventricular zone. So to us, this really suggested that one of the mechanisms of CMV-induced microcephaly is that it may be causing premature maturation of precursors into more mature neuronal elements before migration. So we think that that's a kind of alternative or process that's occurring with neural precursor death that may be causing this disease. So this is hopefully an example of how tissue proteomics can really provide insight into novel disease. So I'm going to end there. Hopefully I showed you some of the ways we're using phenomics or mass spectrometry in this case to study and answer questions about the human brain, how it develops, and how diseases affect it. And of course, there's multiple other ways to do proteomics. There's you can fractionate to get even more proteins, which is something that we're interested in doing. You can further sub-isolate different compartments, which is another initiative in the lab. You can do phosphoproteomics to look for these post-translational modifications, and a number of other techniques. So it's a very, very powerful and dynamic technique that's gaining more and more acceptance and excitement because of its because the technology is now finally caught up to a level where you can do proteomics at the global level. We didn't have time today to talk about some of the use of AI or deep learning for morphomics, but there is a number of lectures, hopefully on NeuroscIQ, that provide some of our recent work on how we use morphomics to also study cell patterns of the brain at a scalable level. So again, I want to thank our funding sources for allowing us to set up our lab and continue growing and do this exciting work. Most of the work was done by Uglisha Juric, a good friend of mine that helps run this amazing team. And of course, a number of new projects that stem from this work have been done by others in our group. So please feel free to rewatch this on our on our YouTube channel, NeuroscIQ, and again, explore some of the other areas that are in the lab. And again, thank you for listening, and please stay tuned for the next. So hopefully you enjoyed that. Obviously, that's one of many studies that we're interested in doing in mapping the proteome of the human brain. So stay tuned for more, and thanks for watching.